Okay, in today's lecture, I will be dealing with mostly that glass ceramic materials for biomedical applications. Particularly, I will address the issue that if one changes the base glass composition, how it will influence microstructure, cytotoxicity and antimicrobial property. Now, there are a number of glass ceramics which are commercially available, but my lecture will mostly focus on machinable fluoromica based glass ceramics. Now, these glass ceramics have particular relevance for dental restorative applications, because these materials are largely machinable, whereas most of the ceramics they are not machinable because they are extremely brittle in nature. Secondly, that these dental materials, now the tooths are of different shape. So, if the materials can be easily machined, then these materials can be given different complex shapes as desired for different types of tooth, and that is the reason that why these materials has received much attention in the biomaterials community. There is other glass ceramic that I must mention, that is 45S5 glass, which was originally discovered by Professor Larry Hench, and these materials have 45% silica, and then you have that different other oxides, and these materials Hench and the co-workers they have actually done the testing up to the clinical stage. Now, this particular machinable fluoromica glass ceramics, they have a particular crystal structure which make them machinable. Now, if you look at this crystal structure here, what you find that you have that potassium atoms which are there at the central region, and this forms a central row of atoms. Then on the top of it potassium atoms, you have that silica tetrahedron, that SiO4 tetrahedron, they are arranged in a particular manner, and then in between the two silica tetrahedron which are there in the top and bottom, you have the magnesium atoms and you have the fluorine ions also. So, this magnesium and fluorine ions, they are also, these ions are also arranged in a symmetric manner, and if you see the top part and bottom part is ideally symmetric to each other. So, because of this kind of particular characteristic crystal structure, what happens during machining operation, these potassium planes they act as a weak cleavage plane, and the materials can be broken along the potassium plane here, and that leads to important characteristics that is known as the high machinability. And the system that it have, this particular fluoromica glass ceramics are observed in a particular system, and this is multi oxide system. And if you see that multi oxide system like SiO2, MgO, Al2O3, K2O, boron oxide and fluorine. Now, boron oxide one has to add some precursor, and this precursor is H3BO3, that is boric acid, and fluorine you cannot add directly, but again fluorine has to be added to this material via precursor, and this precursor is ammonium fluoride, NH4F or CaF2 calcium fluoride. When you synthesize these materials, first one has to obtain the first and primary step for the glass ceramic production, one has to get the base glass composition. Now, how this base glass is obtained? So, like conventional applying the conventional ceramic processing routes, one has to mix these different precursor powders in a desired ratio, then it has to be molten, and during the melting process all these oxides they are mixed with each other, and this mixed oxide melt when it is cooled to room temperature, and when it is cast, and typically the casting is done in a platinum crucible. Why platinum crucible? Because platinum is extremely inert, it does not participate in any kind of chemical reaction with the other oxides. So, because of the noble nature and because of the extremely inert nature of the platinum, these crucibles are used, and where this molten glass is cast in a particular shape, and then after that once this base glass is obtained, then one has to crystallize, or one has to do the crystallization heat treatment, or this is in scientifically known as ceramizing. Now, ceramizing means that you are actually, base glass is essentially amorphous in nature, and then after the crystallization you expect different volume fraction and different shaped crystals to be formed in the base glass matrix, and then the material which is characterized by base glass matrix, where the crystalline ceramic phase are distributed in different volume fraction or different shape, that material is known as the glass ceramic material. People have observed and people have reported in number of multiple systems that typically glass ceramics have better mechanical properties compared to the base glass. And these better mechanical properties has been possible because of the presence of the ceramic phase in the glass matrix. Now, having said this, this particular glass ceramic, that SiO2, MgO, Al2O3, K2O, B2O3F, adopting different heat treatment procedures, people have been able to develop different kind of microstructures. And these microstructures, if you look at the right hand side, there this microstructure has either it can be randomly oriented interlocked sheet like fluorophosphate mica crystals within the fluorine rich alumino-borosilicate glass matrix. So, this glass matrix is essentially alumino-borosilicate glass matrix, and these randomly oriented crystals are what is known as the trisilicate fluoromica crystals. Now, this is that microstructure of type 1, let us say. That another type of microstructure which resembles more like a cabbage type of microstructure, this actually that cabbage shaped morphology is found in with a slightly different base glass composition. Now, this cabbage actually it exactly resembles more like a cabbage vegetables. So, this crystalline phase which forms, it has like cabbage flower or cabbage leaves, and those kind of morphology it has. So, what I am trying to say here, that within the same or almost similar base glass composition, if you adopt different type of heat treatment, in one case you find random crystalline features, in another case you find more characteristic cabbage shaped morphology. So, the message is that one can adopt different heat treatment schedule, and thereby one can obtain different shape and different volume fraction of the crystals in the otherwise amorphous glass matrix. Now, these are the different types of commercially available glass ceramics of similar base glass composition. Now, commercial trade name is Macker, and where Macker the glass ceramic the crystal composition is KMG3AlSi3O10F2. Now, if you can see that these particular crystals, they have a very complicated chemical formula, and these type of crystals essentially reflect that they are essentially generated from the base glass matrix, and this base glass matrix has multiple oxides, and because of the presence of multiple oxides, this crystalline phase also has a very complex phenomena. And you can see here this is like interlock type of mica crystals which are present in an amorphous glass matrix. If you look at this micron bar here, this micron bar is 5 micron. So, typically length of this crystalline phase is roughly around 50 micron or so, whereas width of the crystalline phase is around somewhere 2 to 3 micron. So, roughly the aspect ratio of this kind of crystals may be somewhere around 15 to 20 of aspect ratio. The other things you can see here, that is crystal growth of mica in glass ceramic is essentially diffusion controlled, and this you can see that this is that amorphous phase and where the crystalline phases they are formed, and this is like very high resolution transmission electron micrograph. Now, this to summarize the different properties of the MGC, MGC stands for machinable glass ceramic. They have very high machinability, they are relatively strong, they have good thermal shock resistance and excellent dielectric properties, and they are non-toxic in physiological environment. This property is actually of relevance to biomedical applications, and they have ultra high vacuum compatibility. Now, here you can see this is that 100 plane, that is that atomic arrangement, and you can see that as I said earlier, this potassium plane across which the top and bottom these different silicate arrangement and fluorine and magnesium ions or atoms they are located. So, this potassium plane acts as a more like a weak plane, and that acts as a more cleavage plane here. Now, the reasons for machinability, this is the schematic. Now, suppose you have a crack here, this is like half crack that has been shown, which is propagating from right to left for example, and you have this interlock type of mica crystal. These interlocking mica crystals, now when these cracks will come and they will interact with the interlocking mica crystals, then what will happen? This is like a weak interface. Why it is a weak interface? Because this is the crystal and this is your glass and this is your crystal. Now, glass is an amorphous matrix and crystals is your crystalline morphology. So, this interface typically acts as a more like a weak interface. Now, cracks will initially trying to be getting deflected around this glass matrix interface, but subsequently it would be able to break the crystals and then it will propagate. Now, each time the crack will come and interact with the crystalline phase, some energy will be lost in breaking the crystals and further propagation. Now, if the crack tip energy is lost in this process, then what will happen? The driving force for the crack propagation also will be reduced, and if the driving force is reduced, then brittleness or the more machinability property can be imparted to this kind of glass ceramics. The other things, that is the typical base glass composition. Now, if you see that different type of precursor powders, as I said, the boric acid is used for boron oxide and ammonium fluoride is used for fluorine. Now, these are different weight percent of the different crystalline different powders, they are mixed. Now, after they are mixed, this base glass typically melted at 1500 degrees Celsius. From this 1500 degrees Celsius, you can quench it or you can cast it in platinum crucible. After that, you can do single stage crystallization heat treatment, and this heat treatment either can be temperature variation or can be time variation. In the temperature variation case, we have chosen that 4 different temperature, 1000 degrees Celsius to 1120 degrees Celsius for constant time of 4 hours. And another one is that heat treatment at that time at a constant temperature of 1000 degrees Celsius for different time frame from 8 hours to 24 hours. So, in one case you are actually varying the temperature, another case you are varying the time at a constant temperature, and that is why you are saying in one case time variation sample, in one case we are showing temperature variation samples. Now, this is the XRD plot, which essentially says that at 1000 degrees Celsius 4 hour samples you have that amorphous matrix. Now, when you crystallize it 1040, 1120 and 1000 degrees for 4 hours, what you see, it has a very characteristic fluorophlegopite peak. So, that is a crystalline peak. So, this crystalline peak comes from the crystals that are formed during the heat treatment conditions. And from the FTIR also one can find out that there are different crystal bands, because this is 1000 degrees Celsius 4 hours. You can see that FTIR has only this particular FTIR band. Now, because of the presence of the crystals, these different FTIR bands are formed at different numbers here at different heat treatment conditions. Now, this is the microstructure that was generated after heat treatment at different temperature for constant time frame of 4 hours. Okay. Now, this now initially 1000 degrees Celsius 4 hours, this is the amorphous matrix. Now, at 1124 hours, what you see here, this is the randomly oriented crystals or interlock type of crystals. These crystals is going like this, another crystal is going like that, and that one of the crystals, if you want to see, you will be able to see this is one mica crystals, this is another mica crystals, and they interact. At 1040 degrees Celsius also you will see that different mica crystals, they are growing at different directions. Similar observation has been made at 1080 degrees for 4 hours. So, this is the crystal volume fraction. Based on these ACM images, one can measure the crystal volume fraction, and what you find here at 1080 and 1120 there is no change. So, the crystal volume fraction is roughly around 45%. Whereas, at 1040 degrees Celsius crystal volume fraction is roughly around 58% or so. Now, when you do this heat treatment at different time scale for same temperature, let us say 1000 degrees Celsius, what you see here, you will see essentially the butterfly shaped type of crystals, and these butterfly shaped crystals are what we call spherulitic dendritic type of crystals. They are characteristically formed at 1000 degrees Celsius, and if you increase the soaking time, you can essentially see that somewhere around 25 volume percent to 70 volume percent after soaking for 24 hours at 1000 degrees Celsius, and this is like a systematic increase in volume fraction with time. Now, this microstructure you can see it more clearly here. Now, this is the crystal domain, let us say, and in this crystal domain if you focus that this is a kind of nucleus, and from this nucleus these butterfly crystals, they are growing in different directions, and you can see that a very characteristic crystal morphology here, it is more like a fir tree, or it is more like a tree leaves kind of pattern. Now, you can consider this as a main leaf, and these are like different branches which are formed during this crystallization process. Now, what we believe that during this crystallization process in the base glass, you have this nucleus initially forms, and this when this heat treatment takes place, from this nucleus some solid that this is the nucleus and this is your glass matrix. Nucleus glass interface, some kind of perturbation actually grows, and this perturbation leads to the crystals in different directions, and these crystals are formed, and this main branch of the crystal is what we call primary arm of a dendrite. Now, from this primary arm, the secondary arms they are generated, and these secondary arms they are formed, and if there is sufficient concentration gradient or diffusion field there, from the secondary arm that tertiary arms also can grow. Now, this type of characteristic morphology what we call as a spherulitic dendritic type of growth. Okay. Now, spherulitic type of growth is quite common in many of the glasses, but spherulitic and dendritic growth combination is kind of unique in this particular glass ceramics case. Now, this is what schematically has been shown here, like when you start this heat treatment at 1000 degree Celsius for different time scale, then what you have at initially during these 4 hours, and after that when you go to 8 hour sequence, then all these droplets and this nucleus they form, and from this nucleus you can see that it is growing in different directions or multi-directional growth of the crystals that takes place from this spherical shaped nucleus, and subsequently you will form a typical what we call as characteristic spherulitic dendritic type of morphology. Now, these materials, when we have done this mechanical property characterization, what we have observed that depending on the heat treatment conditions, one can get the hardness something around 5.5 gigapascal, that is the highest hardness, and this 5.5 gigapascal hardness is obtained where at the maximum crystal volume fraction of close to around 60 volume percent. So, higher the volume fraction of the crystals, higher is the hardness data, and roughly the error bars overlap with each other. So, there is no statistically significant difference between these 3 different values of the hardness. Now, we have done the in vitro biomineralization test for these materials, and what we have done, we have prepared the artificial saliva, because these materials are to be used for dental restorative application. So, we do not use the simulated body fluid, but we use the artificial saliva which is present in the oral environment. So that you can simulate the composition which is existing at the oral environment, and this artificial saliva, it has in per 1000 milliliter or 1 liter of the artificial saliva, NaCl is 0.4 gram and KCl is 0.4 gram. As you can see that it is much sodium chloride, potassium chloride, calcium chloride, and there are different sodium containing salts are also present, as well as the urea is 1 gram. Now, when you use this artificial saliva, now what we have found after testing for different 6 weeks for base glass, this is the base glass, before test and after test the glass remains amorphous. But when you do this test for the different other samples before test and after test, you see that there is some texturing effect, because this main crystalline phase somehow vanishes when you do this test for different time scale, and new peaks are actually appearing. Now, new peaks are appearing from different crystal phases here. Now, this is what you can see at 1000 degree Celsius 24 hour sample. After 42 days, you can see these bright contrasting phases as well as this kind of spherical shaped deposits. And now, when you take this EDAX spectra from the spherical shaped crystals, what you see, there is a presence of calcium and phosphorus, significant presence. So, essentially what it tells you that these products are essentially calcium phosphate rich biomineralized products, and these calcium phosphate rich biomineralized products are formed after 6 weeks of SVF immersion. Now, this is what you can see here, the calcium ion concentration, which is most important. At the starting you have this calcium ion concentration. After 7 days and 42 days, depending on what kind of material you are choosing, there is a decrease in the calcium ion concentration in the solution. Now, if there is a decrease in calcium ion concentration in the solution, that should be reflected in the increase in the calcium ion deposition on the material surface, because total volume percentage of the ions should be conserved before test and after test, and that commensurates well with your calcium phosphate rich product formation on the material surface. Now, in the second set of experiments, what we have done, we have changed little bit of the fluorine content in the starting powder, right from 1.1 to 2.5 and 3.85. So, roughly around 4. So, essentially what we are doing, we are changing this fluorine content is 1 to 4 weight%, and accordingly we call it as M1, M2 and M3 glass ceramic composition. And these glass ceramics, when they are melted at 1550 degrees or 2 hours, then you can quench it in water and cast it in platinum crucible. After that, you again remelt this glass ceramic. Why this remelting is done? That is to homogenize the composition, because it has a multi component multi oxide system. If you do not remelt this glass ceramic, all these different oxides, they will not be mixed properly. Now, to ensure that they are mixed properly, you remelt this entire glass ceramic composition, and then you can cast this glass ceramic melt in the platinum. After that, you can anneal these glasses at 610 degrees Celsius. You cut, polish and ceramize the glass ceramics at different temperature, right from 1000 degrees to 1120 degrees Celsius. You can characterize the microstructure mechanical property, and then in vitro property were evaluated. I will go through all these properties one by one. Now, this is the base glass M1 composition, that is the base glass ceramic. Now, if you see the base glass here, this shows essentially amorphous peak, okay, very broad peak, no crystallinity is observed. But the same base glass, when you heat treat at 1000 degrees Celsius for 12 hours, then what you see, very characteristic sharp crystalline peak, and this F stands for fluorophlegophyte. Fluorophlegophyte is the crystal phase that formed when you do the heat treatment in this base glass, and when you do this heat treatment from 1000 degrees Celsius to 1120 degrees Celsius for 12 hours, so the constant heat treatment conditions, these fluorophlegophyte peaks are also much more, intensity is increased, and essentially it says that fluorophlegophyte volume fraction also should increase, and this kind of results is also observed for M2 and M3. So, this is just a representative result that I am showing for M1 glass ceramic. Now, what happens in this FTIR spectrum when we do this FTIR analysis? This is the base glass and this is the glass ceramic. Base glass M1, M2, M3 composition, and this is the glass ceramic M1, M2, M3 composition. Now, you have this SiO Si band, you have this SiO band, and if you have this BO band. Now, BO bands is much more sharper here in this, as well as SiO Si band also. BO band is much more sharper in this base glass, wherever it is much more diffuse and mixed broadened here in this glass ceramic. That is one observation you can make. Other observation is that you have this SiO band is also present, and then there is a combination of the third peak that appears here, which was relatively weak in the case of the base glass. Now, interesting thing that has been observed in this glass ceramics, that one can obtain here much higher volume fraction of the crystals, and this crystalline volume fraction can go up to 58 to 60 up to 70 volume fraction. So, whereas in the earlier case we can obtain only up to 55 or 58%, and most remarkable result that we have got is that hardness of 8 gigapascal. Now, this hardness of 8 gigapascal is very high value as far as the glass ceramic is concerned. Just to give you an example, the steel is mostly fully hardened condition, like when steel is fully martensitic condition, there the hardness is around 7 gigapascal. So, these glass ceramics, this has a hardness which is better than fully hardened martensitic steel also. So, it is really hard, and which is not commonly observed in many of the glass ceramic materials. This is the ACM image of these different Vickers indent that was present on this glass ceramic, and essentially what you see at the 40 gram or 100 gram indent load, you see that this material, that indent has a good shape and then very stable indentation response. Now, this is the summary of all the earlier we investigated glass ceramic materials, and what you see here, that mostly that glass ceramic materials, with some exception of 9.5 gigapascal here, mostly they have a hardness value roughly around 4 to 5 gigapascal or 4 to 6 gigapascal. In our case, we got in the M1 composition 8.2 gigapascal, which is quite good value. In most of the cases, people have not reported the combination of strength and elastic modulus value. In some cases people have measured that bending strength or biaxial strength, and which is roughly around 60 to 200 megapascal. In our case also we got close to 100 megapascal 3-point flexure. In case of the elastic modulus, we have got around 58 megapascal. In other cases, we got 77 to 88 or around close to 90 gigapascal. So, this elastic modulus of 60 gigapascal is good for the bone replacement application, because cortical bone also has a elastic modulus 3 to 80 gigapascal. 3-point flexural strength around 94 or 100 megapascal is also a good value. Hardness is certainly much better than many of the earlier investigated glass ceramic. So, this combination of closely matching elastic modulus as well as high hardness value, that we believe will make this material very viable for biomedical applications. Now, let us look at the microstructure of this material, and what is interesting as far as the microstructure is concerned, that they have what we call as a characteristic envelope type of crystals that are formed in these materials, and this envelope is exactly that latter envelope. You can fold and you can put it, and this each part of this envelope you can see the crystals are formed and they are growing in different directions, and at the boundary of these crystal domains, these two crystals which are growing in two different directions, they come and they meet and their growth is stopped. Now, this is a very characteristic microstructure of these materials. Here you can see that more clear view of this envelope type of crystals. So, this is domain 1 for example, this is domain 2, this is domain 3 and this is domain 4. There are 4 crystal domains, and these are 4 parts of the envelope crystals, and each domain the crystals are growing in different directions for example. Okay. Now, this domain this crystal is growing this direction, this domain this crystal is growing in these directions. Once the growth front they overlap with each other, then the growth of this crystal as well as the growth of this crystal will be stopped. So, they will not grow further. Similar things happen here, this is growing in these directions, this is also growing in these directions. The moment they interact, then the growth of both the crystals they stop, and this stoppage of this growth is taking place at the interface between the two domains. Okay. So, this should be very clear to you. So, this is a very characteristic crystal morphology. Now, if you go back to the introductory slides where I have told you that these glass ceramics, they really provide good opportunity to develop different type of microstructure by changing either the base glass composition or the heat treatment conditions. In this case, you are getting the envelope type of crystals. In other case, I have shown you that you can get that interlocked mica type of crystals. In other case, you have seen that cabbage shaped crystals. So, all different kind of crystal morphology is possible in the similar glass ceramic composition just by adopting different type of heat treatment conditions. So, that is the major message I thought that I want to share with you. Okay. These materials we have done the biomineralization test, and this biomineralization test after 6 weeks, what you see here, this is like calcium phosphate rich layer formation. You can see that calcium and phosphorus speak very clearly, and this calcium phosphate rich dense layer that forms, and this is like spherical aggregates, and these spheres is of 2 micron in size or so, and then you can see they are like they form a continuous layer here, and you can see after 3 weeks also this calcium phosphate rich layer formation starts. Now, as I said earlier that this kind of calcium phosphate rich layer formation is essential for good bioactivity property of these materials. So, once this calcium phosphate rich layer forms, then this material when it is implanted in the body, then it will be quite good idea that this that a biological response of the human tissues to this kind of materials would be much more positive. Now, fluorine ion concentration, this fluorine leaching we have measured at different time period like up to 6 weeks or 8 weeks, and what you see here, the typical fluorine ion is leached less than 0.1 ppm. Now, typically according to the World Health Organization WHO standard, maximum fluorine level in the body that can be tolerated is 1.5 ppm. So, that less than 0.1 ppm for the present set of materials is quite an acceptable value as far as the present materials is concerned. Now, potassium concentration is also decreases with time, like up to week 8 people have done, we have done experiments. Magnesium ion concentration is increases, then becomes stable up to the week 8, but all this, the amount of this leached ion is of the order of some ppm. So, which is quite acceptable for these glass ceramics conditions. Well, we have done some tribological testing on this M2 glass ceramics, and what we see here that after testing against the steel based materials, that are the dry conditions and artificial saliva, you have this COF response, and this COF we do not see any difference. So, in the both the dry as well as SBF condition, COF is roughly 0.7. But interesting thing has been observed when you measure the wear rate, and what you see in the dry condition, that is the normal hatched, that vertical hatch, and the SBF condition, that is the horizontal hatched column, that if you increase the number of cycles or number of test durations or the total test duration, there is a progressive decrease in the wear volume. That means, the wear volume decreases as you increase the total number of cycles. That means, wear resistance is improved as you do more number of cycles or as you increase the test duration. Similar thing has been observed also in the artificial saliva. Here also you can see that this wear volume decreases with increasing number of test cycles. Now, what happens during that, why wear volume decreases or why wear resistance increases? What we have observed, with increased number of cycles, the tribochemical layer forms on the material surface, and these materials now they can be actually, this tribochemical layer is very much protective in nature, and they can prevent the material from further wearing, and that is what is happening here. And you can see this thick tribochemical layer essentially protects the underlying material, and the thickness of this tribochemical layer and protectiveness of this tribochemical layer increases as you increase the total number of test duration or total test duration or total test cycles. You can see after 50,000 fretting cycles that there is tribochemical layer, it starts little bit breakage, but you can see that it is very thick as far as the tribochemical layer is concerned. Now, we have done this in vitro cytocompatibility testing using SaOS2 cell lines. SaOS2 is that osteosarcoma cell lines, human osteosarcoma cell lines, and these cells were cultured for 24 hours on this glass ceramic materials. You can see that phylopody extension of the cells, this is cell number 1, this is cell number 2, and that leads to the cellular breach formations. And also you can see that long phylopody extension. So, that essentially indicates cells are in migratory nature or migratory conditions. And you can see that this typical cell morphology here, and then the cellular network formation also seen on the material surface. Quantification of the cell viability was carried out using MTT assay, and then statistical analysis was also carried out at using post hoc analysis at p less than 0.05 level. Now, we have done the cell culture test for the 2 days, 4 days and 6 days at different time scale. Now, these blue ones are for the 2 days culture, and you see that there is not much difference, only thing this is statistically significant, that is statistically significant for M1 and M3 samples. For 4 days, the green bars, actually you can see the green bars also they are double studies there, and then 6 days their control samples and that M1, M2, M3 they are almost at the similar level. So, essentially all these samples they have similar cell viability as far as the control sample is concerned, all the glass ceramics. ELP activity, that is osteoblast differentiation ability, or this is a phenotype marker of the osteoblast differentiation. And what you see here, that again the post hoc analysis has been carried out, and M1 glass ceramic has the best ELP value, ELP production compared to the M2 and M3 as well as against the control sample. We have done this antimicrobial property evaluation for this glass ceramics using Staphylococcus epidermidis, that is a gram positive bacteria. After 4 hours of incubation, you can see on the control sample large colony formation. This colony formation is still observed in M1 samples. M2 samples it is largely reduced, and M3 we do not see much of this CFU, that colony forming unit, on the material surface. Similar observation has also been made for this gram negative bacteria, that is the E. coli bacteria. This is little bit rod shaped bacteria, and you can see that close colony, very dense colony has been formed of the E. coli bacteria on the control sample. Now, for the M2 sample, there are this colony formation is much negligible, and M3 is much more reduced, whereas M1 also similar antimicrobial properties observed. Quantification was carried out using for the Staphylococcus epidermidis bacteria using UV visible spectrophotometry. This is the mean OD value for this control sample. M1, M2, M3 value, mean OD value is statistically significantly less compared to the control sample. So, what this corroborates well with the our ACM observation. Essentially what it is saying, what it is, what it tells you that both all the 3 glass ceramic materials M1, M2, M3 they have acceptable level of antimicrobial property compared to the control samples.